occur there. So you could see first one, the reaction is a common, the substrate molecule binds with the active site and it carries out the reaction. And the second one, reaction occurs and the product molecules are generated. So when coming to the non-competitive inhibitor, you could see the inhibitor is coming to the active site and it binds to another substances, another site of the enzyme other than the active site. So the inhibitor will bind to another part of the enzyme and it changes the shape of the enzyme and active site, and the substrate will not bind in that and no more product has been released. So for example, cyanide. Cyanide is one such example for competitive inhibitor which binds to the enzyme cytochrome oxidase. So cytochrome oxidase is the enzyme present in the human body where cyanide is a poisonous compound which binds to the cytochrome oxidase. It lacks the oxygen. So it prevents the oxygen from combining with the cytochrome oxidase and it stops the electrons from being passed to the oxygen. So cyanide, it is a lethal poisoning compound that prevents the production of ATP. So ATP is nothing but the energy. So ATP, adenosine triphosphate, is the energy and it is regarded as the energy currency of the cell, so which gives the energy to our cells in cellular respiration. So when cyanide is bind to the cytochrome oxidase, it prevents the oxygen and it stops the ATP molecule to enter into the body. So last point, without ATP, cells cannot perform cellular respiration and which leads to hypoxic condition, hypoxic anoxia and death. So normally all cells of our body, all blood cells of our body, all red blood cells may receive oxygen for its survival. So when oxygen depletion is there, there were no more energy has been produced and which leads to the hypoxic anoxia and death. So without oxygen, the human body leads to death condition. So hypoxic anoxia in the sense very low number or nil oxygen has been found in the red blood cells and there were no more respiration and it leads to death condition. So cyanide is one such example for non-competitive inhibitor. So coming to the uncompetitive inhibitor. So uncompetitive inhibitor, it just differs from the competitive and non-competitive, right? So competitive inhibitor and non-competitive inhibitor is different from the uncompetitive inhibitor. Whereas uncompetitive inhibitor binds to the enzyme substrate complex and not to the free enzyme. So meaning uncompetitive inhibitor binds to after the formation of enzyme substrate complex. So enzyme is there, substrate is there, enzyme substrate complex is formed. In that enzyme substrate complex, the uncompetitive inhibitor will bind so that no product has been released in that. So it requires the formation of enzyme substrate complex. So in uncompetitive inhibition, there are two binding sites. So one is for substrate and one is for inhibitor in the enzyme. So besides this property, enzyme substrate inhibitor complex, it do not form the product because of the conformational change of the enzyme. So coming to the example, so you could see enzyme substrate will formed, enzyme substrate was formed and enzyme substrate complex, the inhibitor is attaching. So enzyme substrate complex and inhibition complex, enzyme substrate inhibition complex is formed. So inhibitor is there and hence no product will come there. So for example, oxalate. So oxalate, it is the uncompetitive inhibitor which inhibits the lactate dehydrogenase. So lactate dehydrogenase is the enzyme. So in that lactate dehydrogenase enzyme, oxalate will go and bind and it is a product of plant metabolism and this inhibitor inhibit the enzymes of lactate dehydrogenase. So lactate dehydrogenase is nothing but it is the enzyme which converts pyruvate into lactate in the glycolysis reaction. So glycolysis reaction, it is the main important form of glucose metabolism in which glucose is converted into pyruvate. So in that byproduct of glucose metabolism pyruvate converted into lactate with the help of lactate dehydrogenase. Okay, so if lactate dehydrogenase enzymes is there, lactate is formed. Lactate is nothing but the lactic acid. So if lactate dehydrogenase enzyme is inhibited with this oxalate inhibitor, lactate does not form, and oxalate can be a potent inhibitor, and it can even increase to the toxicity. So this is the overall summary of the competitor. So in this competitive inhibition, enzyme substrate complex, whereas in enzyme inhibition will bind the substrate, and non-competitive enzyme inhibitor, and in substrate will form enzyme substrate inhibition, and this uncompetitive inhibitor, the inhibitor will come and bind in that enzyme substrate complex. Yes. Reversible inhibitor is over now. As for we have seen a reversible inhibitor, competitive, uncompetitive and non-competitive inhibition. So coming to the irreversible inhibitors, as I said before, irreversible, it cannot be reversed back to the normal stage, and it causes the permanent loss of enzyme activity. So this irreversible inhibitor which inactivates the enzyme by bonding covalently. So as I said before, it just connected with the covalent strong bond. So this inhibitor enzyme is so strong that the inhibition cannot be reversed back by the addition of excess substrate. So one such example of irreversible inhibitor is DIPF, that is diisopropyl fluorophosphate. So this is the inhibitor which prevents it from functioning and binds to the enzyme, and this DIPF, it is a nerve gas. So it irreversibly inhibits the enzyme. So this DIPF mainly attacking the trypsin and chymotrypsin enzyme. So trypsin and chymotrypsin, it is an enzyme particularly found in that small intestine, and it just inhibits the enzyme of acetylcholine esterase, which is involved in the nerve action. So DIPF, it is a nerve gas. So when it is enter into the acetylcholine esterase, it inhibits the nerve action. So obviously the nervous system gets damaged when irreversible inhibitor is bind with that enzyme. So this is the all about reversible and irreversible chemical reaction. So enzyme inhibition, it is a substance which binds to the enzyme and inhibits the chemical reaction. So there are two types, reversible and irreversible. So reversible, it can be reversed, and it is a form of temporary loss, and it is non-covalent bond interaction. In that we have seen competitive, non-competitive and uncompetitive. And what are the examples? Competitive, it mainly competes with the substrate and inhibits the activity, and non-competitive, it just mainly inhibits the other part of the enzyme, not the active site, and inhibits the activity, whereas uncompetitive, it mainly binds with the enzyme substrate complex and inhibit the activity, whereas irreversible inhibitors, it can't be reversed back, and it is the permanent loss, and it is the covalent bond, strong bond interaction, and mainly the diisopropyl fluorophosphate, which is a nerve gas which affects the nervous system damage. So this is the overall about the enzyme inhibition. So I hope you can understand it very well. I can see you all with the another interesting topic. Thank you.